All right, well, good evening and welcome. So, those of you that have been kind of following along, I got the practicals from Monday posted, so the grades were actually really good. We had a huge amount of A's across that, so most people did really well. So, that's excellent. So, we got the practical this evening, so hopefully, obviously, everybody's getting prepared for that. And so we're continuing our discussion about skeletal muscle. So we've been to the nervous system, and we're really at that point of continuation of the signal or that propagation that's going to be coming into the muscle. And on Monday, we spent a good bit of time learning all those players that were involved in the skeletal muscle. So what a muscle fiber is, interchangeable with muscle cell. So we learned all of those things, or at least went over them. And as you can see up here, what we have is really a sarcomere. And this is our primary contractile unit of a skeletal muscle. And we talked about all those players of actin and myosin, or sometimes you'll hear thick and thin filaments, and you somewhat in this depiction of a model can see that. And then when we saw the other model, that the myosin sort of has all these little projections or heads off the end of it, and that's kind of what you can see. So the red is going to be your myosin, and the blue is going to be your actin or thin filament, and their interaction through this sarcomere. We have the different bands, and we'll go through those a little bit again towards the end today. But our real primary task for today is how does this actually work? So we learned about all the players that are involved, but really how does this interact? How does it work? How does it contract and then relax? That's really our big push. And so if you recall through our conversations of previous topics and subject matter, that we will basically go through a step-by-step process. So there's about 18 steps for a muscle to contract and then to relax. And so we'll go through those this evening, and we'll go through it a time or two. If it's the first time you've heard this, obviously it's almost like a different language as we spoke a little bit before. But we'll kind of talk briefly about sort of a few things. And this slide talks a lot about the contractibility of this sarcomere unit, and that's really what you see up here. And it runs off of a theory called the sliding filament theory that really wasn't around until I think around the 50s and so forth. And as we said, if you remember, this is the Z line to Z line. That represents the actual sarcomere. And this model here shows the sarcomere at rest. And that's where we spoke about, so we have in the middle this A band, and that A band is where we see ultimately the thick filament, and its overlap into actin. And then over on the other side, this has sarcomere contraction of the slide. So these will actually shorten. So almost like if you put your fingers together such as this, that they have to touch each other so they can sort of ratchet themselves closed and so forth. And we'll go through each one of those as we go. And so this is an individual sarcomere at rest and also at contraction. And then we can see down here that this is really a long myofibril during contraction. So at the end of this, there's going to be multiple cells, and you can see that interaction there all along through it. Each one of these that you see down through here is a sarcomere unit. And then as you can see, this is at rest, and this is in contraction. So you can see it doesn't shorten what you would think. If you thought about it, we just got through all the muscles in lab, but if you thought about, contracting your bicep, that you would think that these are bigger units moving bigger lengths, but we're talking really small areas. And as I said before, on the task coming up next week, there's a few questions that specifically talk about H bands and what's happening during that contraction. Which ones are staying the same, which ones are shortening, and so forth. So if we take a look down through these primary 4 steps on our slide set here, so we can see skeletal muscle fiber, when it contracts, it says the H zones and the I bands will get smaller. And so if we go up here and rationalize, well, why is that? So if you remember, the H band is really in the middle, so that is from only where there's no actin. So from here to here, no actin. But in a contracted phase, that H band is about a third of its size. So it's actually pulled that actin and myosin together as we go. It also says here that the I band will get smaller. So the I band is really primarily between the Z line and the edge where we start the overlap. So you can see the little green, almost like that spring we spoke about. That's that titin protein. And so as we take a look over on this one in a contractile state, we can see that that I band is very small. So this particular sarcomere is contracted to its maximum. It can't contract anymore because there's no potential play for it to go. They can't get any closer. They've already contracted and squeezed in together as close as they can get. So in that state, H and I bands get a little bit smaller. The zones of overlap get larger. And so that makes sense because as the two pull together, we can actually see this area to here in the contractile state is much longer than here, about half in nature. And then the Z lines move closer together. And that makes good sense, right? So if we looked at what a sarcomere looks like, it's from Z line to Z line. And so as the filament gets shorter, the whole product gets shorter. And you can see that we've shortened that area from, maybe by a third or so. And then the width of the A band remains the same or constant. And you might ask, well, what is the A band? Well, if you recall, that in essence is just where the thick filament or myosin lives. So it's not going to change. So it's somewhat of a little bit of alphabet soup. It's just a little bit of memorization to kind of get the bands down and which ones are doing what, but it's imperative that you understand which one is actually shortening, staying the same, and getting longer. As we kind of move on just a smidge further, this tells us just a little bit more about that process as we spoke about. So the sliding filament theory basically says the thin filaments are sliding towards the center and each sarcomere along the thick filaments. It's ultimately what we just spoke about. The myofibrils will shorten, causing the muscle fiber itself to shorten. And those are those two pieces that we saw before about those interactions with that myofibril in its relaxed state. And then on the bottom, we see it in its contractile state. This theory, you don't have to know this, but it hasn't really been around a long time. It's on this slide, and it's been on mine for a long time, even since I started here. But this gentleman, I just like to say his name, I think, because it's kind of fun to say. But Hugh Huxley in 1954 really came up with this. So not really that long ago, if you really thought about it. So we're really, now we're pushing 75 years or so. But you would have thought that we would have understood how a muscle contracted much earlier than that. But really, that's really when we came about it. And so you don't have to know this. We just put on here for sort of to round things out. But he found out that we had actin and myosin, how their interaction is. And I'm sure he had a team of folks helping. But in relaxed muscle cells, overlapping and thick and thin is much smaller. So that the filaments sort of slide and overlap. And that's really the super simplistic way to think about it. So we're going to take a deep dive over the next maybe 15 slides to really take a look at how does this thing work. So this is really the first slide, and you have to think about this from a neuro perspective as well, because here's our motor neuron. And so we covered neuro already, but as you remember, this motor neuron lives where? So this guy right here is going to be in the spinal cord. It's going to be in that gray matter of that spinal cord. And this axon is going to be coming all the way out to the muscle, to what we consider a muscle end plate. So when we get to the muscle end plate, these are the synapse area that we're speaking of. So these are our primary players. So this blue area here is actually the end of the axon as it goes down to that motor end plate. And the motor end plate has a synapse or synaptic cleft, the same as what we spoke about pre and post synaptic neurons. It's the same idea. So we have to have a chemical to get across that synapse to stimulate and make the muscle contract. So if we're going to go back to those terms about graded potentials, action potentials, those guys again, and how it's going to stimulate this to actually contract, then how do we get it to relax again? So as you can see here, we'll kind of go through some of the bullet points, but we're primarily looking at, we know that this is all under neuro control. If you remember, anytime you hear that somatic, we should be thinking skeletal muscle, somatic skeletal muscle. And it's voluntarily controlled, so your mind, your brain has to tell your muscles to move, to contract, to hold tension. So we a lot of times think primarily only about muscle contractions, to move, to pick things up. But as we spoke a little bit on Monday, as you sit in the chair, as I stand here, there are certain muscle groups that are just going to be contracted to tension, just to sort of hold it there, sort of help so you can stand up, so we can balance. So if you can sit in the chair, if you didn't have those muscles at tension, then you would fall out of the chair. So these communications occur at the neuromuscular junction, or NMJ. So you'll see that a lot of times. And so that's what we're referring to here, that we've blown up on the model over here. And you can see all of our players here. We have a synaptic terminal. And if you remember, we knew there were a lot of names for that thing. Synaptic knob, there's a terminal bouton, there's all kinds of names, but it's basically the synapse, or the end there, the terminal. And then inside, you can see our guys. Those are our vesicles full of neurotransmitter, motor neuroplate there. Then we have some of these other parts that we start speaking about, and we'll see this more and more, but if you remember that sarcoplasmic reticulum, very similar to endoplasmic reticulum, but surrounds those myofibrils within there, and they are primarily going to be stimulated to release calcium. So each muscle fiber is controlled by a neuron at a single neuromuscular junction. So this gives us the ability to either recruit a large amount of muscle or just a little tiny bit. And we used that example before. Sometimes if you try to pick something up and you can't pick it up, and you kind of try again, take a deep breath a lot of times, blow out, and then you're able to pick up a heavier weight. Because your mind is always calculating how much does that weigh? How many muscle fibers do we need to stimulate to pick something up? We use that example about the soda can, because we can't see in a soda can, right? So if we've had a soda or whatever beverage you like, and you've had, most of it, just leaving a little bit in the bottom, and somebody brings you another one and opens it and puts it side by side. And we thought, that we're going to pick up in your mind, we was like, well, I didn't want that old one, and it's kind of flat, I'm going to get the new one, and you mix them up in your mind, and you pick up the one that's empty, you pick it up off the table. Everybody's done that sometime in their life. When they're going to pick something up, and it's just like, you spilled everywhere. Because your mind has recruited way more muscle fibers than it needed, because it thought maybe this is heavier than it was. And we'll spend a little bit of time talking about that on Monday, about how that occurs, but we're going to take a look at all of these processes about how this thing actually works. So it tells us a single axon, so remember our axon, and this is myelinated. So if you remember myelinated, so it's going to jump down through there, saltatory, just between those nodes of Ranvier. So that signal is going to be coming down really quickly. And at the end of the branch is that synaptic terminus. And if you remember, there's our acetylcholine. Remember, acetylcholine, or ACH, is the most common neurotransmitter that we have. We spoke Monday about a few others and where they live and so forth, but out in the muscle, acetylcholine is king, so to speak. Now, as we continue our conversation, so this is blown up at that synapse. So as you can see, this is that synapse, or the synaptic cleft. So the brown area is all of the synapse. So if you recall, the synapse is there so we can excite a muscle or excite whatever is past that synapse. And yet again, we can have it at rest. And there's only a couple instances of where they touch, like the eyes. Remember we spoke about that. So we have to have a mechanism to get across there so we can have that message or that impulse, electrical impulse, the voltage coming down, to be able to cross over that synapse to basically stimulate this down here. So the gray, or I guess that's kind of a vanilla color, I don't know. Anyway, so it's actually going to be what is the muscle in our models. So it tells us over here that the motor end plate, the region at the sarcolemma that contains membrane receptors for ACH or acetylcholine. So if you remember, the sarcolemma is the plasma membrane of a muscle cell. And that's all we're talking about. And that's all of this here. So there's a couple other players on here. We know that the red are vesicles containing that acetylcholine. Out here in the synapse itself, we see the little half moons. And we'll use those half moons, and those would be acetylcholine esterases. So that's why it's ACHE. And so it ends in ASE, so that means it is an enzyme, correct? So when we need it, that will come and pick up the acetylcholine, break it apart, and it'll be reabsorbed. Now, what you see also in this particular model are these folds. They're called junctional folds, and you'll see these little purple areas, and the purple areas would be ligand receptors. So they're chemical receptors specific to acetylcholine. So that's what they are. And these folds, in essence, just give us more surface area. So if you basically took a piece of paper and folded it up like an accordion, it would take up less space, but we would still have the same amount of surface area in a little tiny area. And that's really what these folds do, and they allow more of the receptors for acetylcholine. So these are some of our players that we've got going on here. And as we continue a little bit, these are those terms that we need to get in your mind to basically be able to understand some of the questioning. So these are just sort of some behavioral skeletal muscle fibers. So we have 4 primary phases. So the first one is excitation. So it's basically the process in which the nerve action potentials lead to muscle action potentials. So this tells us that the nerve action potential is coming down the axon into that synapse. And then how are we getting it across? We'll spend some time about that, from a graded to an action potential. Then we're going to have excitation and contraction, which is considered coupling. And that's really where we're going to see that interaction of actin and myosin. And so that's really what we're saying there. So it says the events that link the action potentials on the sarcolemma to activation of the myofilaments, therefore getting them ready to contract. So we're kind of getting them prepared. So how this works, we don't want to get the cart too far before the horse to go through our steps, but that action potential hits that sarcolemma and it propagates. If you remember that word, propagate, so it's going to spread across that muscle fiber, almost like if we had leaves or paper on the floor and you threw a match in the middle, from that synapse it's going to sort of spread out and start to stimulate that muscle. The contraction phase is really basically when that muscle fiber is going to develop tension or shorten, I guess you would say. So do we just need to sort of hold in place? So if you kind of think back when we were studying muscles, and obviously we somewhat still are in the lab, is most of our muscles are sort of analogous. So we have bicep in the front, tricep in the back. And so as one contracts, the other might hold tension. So, if you kind of do this, you can touch back here. It's not contracted, but there's a little bit of something going on there, right? So it's just helping to hold that in place. So we sort of get that. And so the next one is the relaxation point. It's like when the stimulation ends, when that impulse does no longer come from that nervous system, what truly happens at that point? The muscle fiber relaxes and returns to its resting length. So we are going to go through literally a step-by-step process of how this occurs and how we're going to basically see it go through all four of these. And we're going to start basically right now. And this particular slide is going to take a look at excitation, or we're basically exciting it. We're basically having that particular impulse go on. So if we take a look at all of our players, so here is basically our motor nerve. Here's our sarcolemma of our muscle cell. And we're going to have the arrival of a nerve signal. So the red is going to indicate the impulse. So if you recall, so what's happening as this comes down, that's sort of our, hey, we're going to hit that, we got to get to that -60, then we're going to go all the way up to +30, start back down. That's sort of what we're doing. We're continuing that process as we come down through here. So if we look at our bullet points, it tells us that the nerve signal is going to open voltage-gated calcium channels in the synaptic knob. So that's important. So we spoke about gated voltage channels in the neuro, right? That's where we had the activation gate, inactivation gate for sodium. We also had an activation gate for potassium. And they opened and closed based upon a millivolt change, right? So at -60, we have an open. That's what causes the depolarization. And we're coming back down the other side. We start to see some those gates swapping back around. So when the nerve signal comes down here, it's depolarizing all the way down through here. When it gets to the knob, the voltage change opens this gate. There's more than one. Obviously, this is very simplistic and somewhat in its nature, but there's going to be multiple calcium channels here. And when that opens, calcium is going to rush into this synaptic knob. So that's really what we say in bullet point 2. Calcium enters the knob and stimulates the release of acetylcholine from the synaptic vesicles inside the synaptic cleft. So what happens? That sodium or that calcium entering into the cells is a stimulation for the vesicles to go to the surface, open or excise their material, which is acetylcholine, into the synapse. And you can see that. So there's, in our model, we got 3 primary vesicles opening and releasing the little green acetylcholine. Now, the purple and the folds, junctional folds, are representing the acetylcholine receptors. So that's what we have blown up here. And we need 2 molecules, as you see here, or chemicals, ligand. It's all interchangeable, but we're specific to acetylcholine. And it's going to diffuse across and open those receptors. So that is step 1 and step 2 of the excitation process. So if you think about it, we're just picking up where we stopped our conversation with neuro, right? So we were all the way to the synapse. We talked about the synapse. We talked about who's in there. And so as our message comes down the axon, enters our synaptic knob, whatever you like to call it. As you know, we got the Bhutan, lots of things that we can call it. But that calcium is going to rush into that knob. It's going to cause the release of acetylcholine. The acetylcholine then will go out, find the receptors, open them, and we'll see what happens next. So slide 2 of our process here, as I mentioned briefly there, but so it takes 2 acetylcholine molecules to bind to each receptor to open it. So this is very similar to what we spoke about way back in the beginning, right? We had to have 2 things to open that particular channel. And so now we see our good friend sodium, right? Sodium is our primary way that we can change voltage. Way back when we spoke about the chemical and electrical properties of sodium and potassium, that we know that sodium is the one that's responsible for voltage, or the very quick ability to excite that membrane. We know that our potassium can change the millivolts, but it doesn't have that power to change the other way. So what happens? The sodium enters, shifting the membrane potential from minus 90 to plus 75. So what have we done? We're starting the excitation phase. Then we have some potassium is going to exit, and our potential returns back to minus 90 millivolts. So that's sort of the end. The quick voltage shift is called an in-plate potential. So the ability for that sodium to rush across that membrane through our channels causes that voltage shift, and we, at the in-plate potential, basically we're now getting to the point where we're exciting that. So number 3 is we had our acetylcholine bind to the receptor here and here, our 2 pieces of the puzzle. And then we have our movement of our sodium in and potassium out. Therefore, stimulating our sarcolemma. So how does this continue on? So as you can see, this area here is the plasma membrane of the synaptic knob is here, right? This is our sarcolemma, basically a phospholipid bilayer in essence. Here's our receptor with our 2 here. We still have some free floaters out here in the synaptic cleft. But as you can see over here, so we have sort of lit the fire, so to speak, here. We've excited the membrane. We've depolarized it. And then over on the membrane itself, there are voltage-regulated ion gates. And as that voltage change, sodium in, potassium out, the message continues on across the whole, that particular area that synapse is responsible for. And that's what you see here in our bullet point. So this is number 5. We're going to we're opening the voltage-regulated ion gates. We're creating action potential. So remember, action potential is message over a longer period, right? Gradient potentials are shorter, as we spoke. So it tells us the voltage change in the in-plate region opens nearby voltage-gated channels, producing an action potential that spreads over the muscle surface. So it's almost like we had that leaf pile and we tossed a match in the middle. So the first little match area is our opening of those channels. And then we're going to spread that message and that voltage all around that particular muscle. Okay? And then we'll kind of continue on our path. So as we get here, so that was the excitation part of it, right? So now, what's our next phase? So if you remember, it was contraction and coupling. So we're just preparing all of our folks here. So if you remember from Monday's conversation that we had that sarcoplasmic reticulum sort of around these myofibrils, right? And so we had the T-tubule that was coming in off the surface. It was a little hull. And as you can see, we have this triad. And so the triad, if you remember, is the T-tubule in the middle, and then it's terminal cisternae on both sides. And the blue, the sarcoplasmic reticulum, is full of calcium. And that's really important to us. We have to have calcium, and lots of calcium. So what happens, if you've ever seen Star Wars, it's the Return of the Jedi. You know, everybody hopefully has ever seen that movie. It always reminds me of this, I guess. I was a big Star Wars fan, years ago. But it's almost like, hey, we've got that rocket, we got to shoot it in that little hole on the Death Star. Luke comes down through. Nobody remembers, right? I'm old, so that's okay. So that message. So here's our synapse. We've already excited this area. And these voltage channels are pushing that action potential, and they go right into T-tubules. Wherever there's a T-tubule, they go in. That voltage, that change goes in all those T-tubules. When that occurs, so it's going to tell us here, the action potential spreads down the T-tubules, just like that, goes right in the middle. headed down the middle. That opens voltage-gated ion channels in the T tubules, and we have calcium channels that are going to open. The calcium leaves the sarcoplasmic reticulum and enters the cytosol. So that's the cytoplasm of that muscle cell. Okay? So that is important. So we're getting to the point where we're close to contraction, but we're not quite. You got to remember, this is happening very fast, based on our conversation. So it goes in, it stimulates voltage channels inside there. That's going to cause the calcium to be released from that sarcoplasmic reticulum, or primarily that terminal cisternae, and it's going to basically start to wash in and over that myofibril. Okay?